Assalamu alaikum. Hello guys, welcome back to my channel. I prepared this time to explain in a different way of course, histology basics. Today, now I will explain part 2 of connective tissue cells, which are free cells. As I explained about, those are not wrong, but the computer, I don't know what's going on with it. I should give spaces, I don't know, capitals, I don't care about those so much. I will explain about part 2, which are free cells. So the free cells, we have 4 major types of them, and I will explain about them now. Okay, the first one we have adipose cells or adipocytes. They are even called adipocytes. We have 2 types of them. So number 1A is white and brown. So they look like each other simply, but this is the simple appearance of lipid or adipocyte under microscope. We have so much names for this. We can say adipose cells, we can say adipocyte, we can say if they are a group of adipocytes, we can call them adipose tissue too. So if we come to this is the membrane. Okay, they are round and they are recognized very easily. So this one is the nucleus. This one stains basophilic, but not very strongly basophilic. This one is the nucleus. So this is just the membrane. Okay, another cell. This one is nucleus, membrane. Okay, another nucleus here. This is a slide stained by H and E, hematoxylin and eosin. Okay, the second cell, we know that the function of adipocytes are heat regulation and fat storage in our body. So number 2, we have mast cells. Mast cells, we can call them immune cells. They are a type of immune cells. They are large, polymorphic, spherical or ovoid cells. So they may look like circular, like that, or oval in shape, but mostly circular, spherical and circular. Okay. Located, so the location is in the loose connective tissue. What we mean by loose connective tissue means they are not attached together, just as the epithelial tissue. Loose connective tissue, especially of the skin, so the skin, loose connective tissue, intestine, and around the blood vessels. In my other videos, I explained about some slides, which one of them is the intestine that you can look. I explained about the epithelial tissue, presence of lymphocyte, and connective tissue. Blood vessels and around the blood vessels. LM means light microscopy. Light microscopy of the mast cell. If we come to the light microscopy of mast cell, this is a light microscopy of mast cell. We will not, I will not tell you if this is normal, abnormal, something pathogenic. I will just show you what's a mast cell simply. So, they are filled with metachromatic granules. Granules are those dots. Those dots or spots are called granules. Those spots around in the cytoplasm are called granules. They take different color from the dye. What we mean, if they are stained with toluidine blue, they take red color. This is known as metachromasia. So what's metachromasia? For example, you stain this by using toluidine blue stain with the colors blue. What will they appear like? They will appear in another color opposite of the stain of the dye. So what will we see? We will see something, just for example, red if it's stained by toluidine blue. So this is called metachromasia, okay, or metachromasia, though there's no difference between the pronunciation. So what's the function? What does mast cells do in our body? In allergic reactions, they remove chemical mediators. They remove chemical mediators in allergic reactions. The granules of mast cell contain histamine and heparin with ECFA. So ECFA, eosinophilic chemotaxic factor for anaphylaxis. Okay, this is the long form. So number 3, the surface of the mast cell contains specific receptors for immunoglobulin E. So in those surface, the outer membrane, we have receptors that are specific for immunoglobulin E, or we can say IgE. So we would come to the third type of cell, which is very important to know. Those two cells, recognizing mast cell may be sometimes easy, but plasma cell and melanocytes that I will explain about are sometimes hard. So, guys, so plasma cells, we know what's plasma. So what's the difference between plasma and serum? Let's go get out a little bit and get some extra facts. So plasma has, we know that the plasma has clotting factors which are inhibited by an anticoagulant. If we see a sample of plasma, but serum, the clotting factors aren't inhibited. If we get a tube, for example, that contains serum and plasma, someone asks you what's the difference between plasma and serum. That's what will you say to the person who asks you. So plasma cells, they are spherical, oval in shape, or sometimes pear-shaped. Okay, and it has a spherical eccentric nucleus. What means eccentric? Means they are nearly attached to the membrane. They aren't in the middle. They aren't here. No, they are attached to the membrane. But some cells have nucleus in the middle of the cell. So they are eccentric nucleus with condensed nuclear chromatin. So this is the nuclear chromatin. They appear condensed. Those one in the nucleus. Those are the chromatin. Okay, this cell is a neutrophil, guys. This cell is a neutrophil that I will explain about. I will make a special video about blood and white leukocytes. This one is called a neutrophil. How do we know it's a neutrophil? We have bilobed nucleus, okay, and yeah, we have bilobed basophilic nucleus. These cells are red blood cell RBCs, okay. RBCs, and we know that neutrophils are types of leukocytes or YBCs. This one is a platelet. This one is a platelet. Platelet has a role in blood clotting, so this one is a platelet. They stain basophilic. This is platelet. So this is plasma. The location of plasma? They are in the connective tissue, especially those of female reproductive system. So those of female reproductive system, and in the respiratory system. So means in the respiratory system we can find plasma cells. There are I will explain about the types of tumors that we may get pathology, and they have strongly basophilic cytoplasm. We know that the cytoplasm is basophilic, as you see here, and with a prominent negative Golgi apparatus. So what's a Golgi appearance or a Golgi apparatus image? So what's Golgi apparatus? You see this white place, this white area. This is the Golgi. This is a negative Golgi. This is called negative Golgi. Okay? This is called negative Golgi. In past videos, I showed, for example, some cells. If you stain cells with the silver stain, you will see clearly negative Golgi appearance. Okay, let's not forget, for lipids, we can use special stains that are called osmium stains. Those are important to know. The last type of cell, so this slide, let's give a review for this slide. This slide is important. You will need it. All your vet or any medical school you will need it for which part are you? Are you a cytologist, a microbiologist, a physics, or any part, even if you are a bachelor degree, if you are studying any medical college, you will need the, sorry, my battery is low. You will need the slide. So this is red blood cell. Those are the platelets. This is the neutrophil, a type of white blood cell, and this is the plasma cell. with a negative Golgi appearance here, and this is another plasma cell. Okay, and this is the ground substance. Okay, so melanocytes are found where? They are derived from the neural crest. They have large pigmented cells with numerous long branching processes, and the locations they are found in the meninges. So where are the meninges? They are in the brain. Okay, they are located in the brain, in the behind part of the brain. You will study it in anatomy. Okay, and the epidermis, which is under the which is the in the last part of the epidermis, and uterine of sheep, crankles, and iris, and you can find it in the choroid too. So here are the melanocytes. Melanins simply give skin color. Okay, this one that have, they may appear blue, sometimes black, or brown. It depends on the slide that you will see. So this is the keratin layer. This is a keratinized tissue. In skins we have keratinized. This is the epithelia. Okay, this is the epithelia. We have the basal cells down part, and we have the melanocytes. Okay, so this is the layer. I will explain about those layers. Stratum corneum, stratum lucidum. I will explain about all of them. Here we have another slide which shows those melanocytes. This one is a very nice melanocyte with a nucleus, as you see. This is a nice melanocyte. This is another melanocyte. This is this melanocyte. I like it so much because it's so nice. As you see, this is the pigment appearance, and this is the nucleus. So those types of cells may be tumors, may be abnormal. For example, we have lipoma or lipid cell tumors, mast cell tumors. We have them, plasma cell tumors, and melanin tumors, or melanoma. We can say melanoma. So every normal cell that you study, it has a pathogenic form. Okay, so simply it's just like that. So thank you guys, hope you get benefit, and don't forget subscribing, of course.